thanks for joining us, everybody. We're lucky to have with us today Alex Marson. He's super in demand. We had to ask to get him on the calendar about a year ago. He's director of the Gladstone UCSF Institute of Genomic Immunology and a senior investigator at Gladstone, as well as an associate professor at UCSF in the departments of medicine and microbiology and immunology. And Alex is interested in how DNA controls the behavior of cells in the human immune system, not the mouse immune system, the human immune system. And he uses the power of CRISPR to genetically engineer cells with the goal of fighting cancer, autoimmune diseases, and infectious diseases. Alex completed his undergrad at Harvard and earned an MPhil in biological sciences from Cambridge. I'm sure there's a story there too. Earned a PhD at Whitehead Institute at MIT, where he worked with Rick Young and Rudolf Jenisch on transcriptional control of regulatory T cells and embryonic stem cells. Great to have you here, Alex.
Thank you for that really nice introduction. And thanks for having me today. I've been really sorry it's taken a little while to schedule it, but I've been looking forward to this for a long time. And I hope it sparks some discussion.
Great. So hopefully you can speak for about 40 minutes and we'll take some Q&A from the audience toward the end.
Sure, that sounds great.
Great.
So I'll tell you a little bit about the work that we're doing in my lab at the Gladstone UCSF Institute of Genomic Immunology. And broadly, we're interested in understanding how DNA sequences control the behavior of cells in the human immune system, with a particular focus on T cells. And we have put a lot of effort into using genome editing tools to really be able to go in and rewrite these DNA sequences to understand how they control cell functions, and actually to think about reprogramming cells to transform them into cellular medicines that can treat a range of different human diseases. So before I get into the details, here are disclosures that I'm supposed to share with you. Actually, you were just asking me before we let the participants in, how did I first get introduced to CRISPR? And I thought I'd start actually by talking about 2012, which happened to be the year that I came to UCSF to finish up my clinical training in infectious disease. And I was just starting to think about what my lab at UCSF would actually do. And two things happened in 2012 that had really a profound influence on the field, but also on my thoughts and on the course of research that I took. One of them happened in Philadelphia. Emily Whitehead was the first pediatric patient to get treated with CAR T cells. And she was treated by, the study was led by Carl June. And the results were really miraculous. She went from being almost sent home on hospice for an incurable type of cancer to now remaining cancer-free all these years later. So this was really a long-term response, maybe hopefully a cure for her type of cancer with a genetically engineered cell. And so I think this really woke me up to the idea that genetically engineered cells could be a way of effectively treating human disease. And the same year, 2012, this paper came out that really rocked all of biology from Emmanuel Charpentier and Jennifer Doudna, that showed that CRISPR could be repurposed as a tool to very flexibly edit DNA sequences. And the thought started to emerge, well, what if these technologies really could converge? And we'd have a tool to take cells and actually rewrite their DNA sequences to build more effective cellular medicines. That's been the basis for much of my work over the past decade. So briefly, just to introduce what CAR T cells are. Again, this is probably one of the most developed form of genetically engineered cell therapies. Patients with certain types of cancer can go and give blood samples. Cells are taken outside of the body and genetically engineered. In the first generation of CAR T cells, including ones that are now FDA approved, shown here on the right, this genetic modification happens with viral vectors, lentivirus or retrovirus, that insert transgenes for artificial genetic sequences into the T cell genome. They insert a gene for a chimeric antigen receptor, which artificially directs the antigen specificity of those T cells to recognize something on the patient's cancer. And then these genetically engineered chimeric antigen receptor or CAR T cells can be reinfused into the patient. And for certain types of blood cancers, the results have really been astoundingly positive. and as I said, have led to a number of FDA-approved therapies. There's still a lot of work that needs to be done. And so even for leukemias and lymphomas that sometimes respond, we need to figure out why not all patients are responding, or why in many cases there are relapses. So we'd like to make more durable, more effective therapies for blood cancers, but for solid tumors, CAR T cells have been largely ineffective. So we also have to figure out how to expand the reach of these types of cell therapies to a broader range of cancers, including many causes of major cancer mortality. And we'd like to really be able to expand these types of cell therapies, not just to cancer, but to autoimmune diseases, to infectious diseases, and other types of human diseases. And we remain optimistic that that is all going to be possible, that we are just seeing the tip of the iceberg of what genetically engineered cells are capable of. And what gives us hope is that we can really now move beyond these sort of first generation viral vectors, which have been effective, but which have laborious manufacturing requirements of going through viral manufacturing steps, and also which the lentivirus and retrovirus don't insert transgenes at targeted places in the genome, really limiting the types of genetic edits that you can do. Now with CRISPR, all of a sudden we have a tool where if we can deploy it in human T cells, we can actually pick a specific site in the genome and fix individual mutations, but we can also remove genes in the genome that are limiting the function of cells, checkpoints and other things that we could potentially release to make more powerful cell therapies. Or we can even imagine taking a targeted site in the genome and adding a new sequence, or replacing a long stretch of the endogenous sequence to insert a new synthetic program at a defined site in the genome to really make cells behave how we want. So just to give you a flavor of one of the genetic programs that there's been a lot of effort on is rewriting antigen specificity. I mentioned, so this is writing what a T cell will recognize and target. I mentioned that this can be done with chimeric antigen receptors, or CARs. It could also be done with the cell's own T cell receptor, the endogenous receptor that determines specificity. And a number of years ago, Justin Acomb and Michelle Sadelain showed that there's actually a benefit if you could potentially put a CAR into a defined site in the genome, in particular, put it under the endogenous gene regulation that regulates the cell's natural T cell receptor. If you could put the CAR under those gene regulatory elements, you could actually, in preclinical models, make better CARs, more durable CARs. So we've put a lot of effort into developing a very easy to use, non-viral way of using CRISPR to do targeted rewriting of antigen specificity, either by going into the T cell receptor locus and replacing it with a CAR sequence at that site, or directly replacing it with a TCR sequence. And this allows us to start making T cells that can recognize cancer antigens, either with CARs or TCRs. And the way we do this is by electroporating T cells with CRISPR, with Cas9 ribonucleoprotein, Cas9 nuclease plus a guide RNA, along with, at the same time, we introduce extra DNA with homology arms targeting the cut site of that CRISPR. And this has come a long way. We now have this working extremely efficiently. So this is after a long progression, starting many years ago with a great collaboration with Jennifer Doudna. Now in the most recent work led by Brian Shai, which was published last year, we now have a non-viral system that is achievable at clinical manufacturing scale, super high efficiency and high cell yields, showing that we can get insertion of a CAR sequence. This is a BCMA CAR targeting a myeloma antigen at very high efficiencies. And so this is a very flexible tool to rewrite antigen specificity. But we know that that is really only a small part of what we need to be able to do to make effective cell therapies. Even if we can make an antigen-specific T cell that recognizes something in a solid tumor, either with a CAR or TCR, those T cells will face a number of challenges that can limit their function. They will be chronically stimulated by many cancer cells as they try to clear a solid tumor. There's checkpoints that they'll encounter, like PD-L1. There's suppressive metabolites and cytokines that can accumulate inside a tumor microenvironment, like adenosine and TGF-beta, and there's suppressive cells. So we recognize that in addition to putting in antigen specificity, we really like to figure out how can we use CRISPR to make other genetic modifications that will confer a target profile on these engineered cells. And some of the things that we've thought a lot about are, well, we'd like the T cell to the engineered T cell to proliferate when it finds its target. So you get more of these T cells. We'd like it to resist all these modes of suppression that I just talked about. We'd like it to be durable over time, so you get long-lasting responses. We'd like it to remain sensitive, even if cancers start to downregulate their antigen density. So we'd like these to be sensitive therapies. And through all these states, we'd like them to not only endure, but we'd like them to endure, the T cells to endure in a state that's compatible with them having the properties necessary to continue to clear tumors. So how can we design cells with these properties? Well, we have really turned to the power of forward genetics. And so this is something that CRISPR has been extraordinarily powerful for, not just manufacturing a cell, but actually as a discovery tool to discover what are the genetic edits. And the way we do this is we make many, many different genetic modifications in a population of primary human T cells. And then we essentially race all those cells with different modifications against each other and pick out the winners that for different properties that we care about, so that we can accelerate the discovery of which genetic modifications will confer desirable behaviors into these cell therapies. And so I'm going to talk for the remainder of the time today about giving you vignettes of these different tools and how they're teaching us really fundamental lessons about T cells are wired through their DNA, and also giving us new targets for gene modification in cell therapies. So first, a couple of years ago, we adapted tools to knock out every gene in the genome in a population of T cells. We have also more recently, just last year, figured out how not only to knock out, but actually to transcriptionally activate every gene in the genome in a different population of cells. And what you end up with in either of these situations is a mixed pool of cells, where each cell is not only has a different perturbation, but which can be easily tracked by a barcode that we've introduced into the cells. And then a complementary approach that we've invested in a lot is not going throughout the genome, but picking one targeted site in the genome, and now knocking in a different DNA sequence into each cell in the population. And these knock-in templates include barcodes. Again, you end up with different perturbations introduced into each cell with different barcodes that allow you to track the behaviors of these cells. Once we have, through any of these perturbation technologies, a mixed population of cells that are barcoded, we can race them against each other into different conditions and see which ones proliferate the most, which ones have a survival benefit. We can actually sort them based on, for example, FACS characteristics to pick out cells that are in a particular state and see which modifications have given them that state. Or we can actually look even more broadly at state, and each of these perturbation technologies are now compatible with single-cell RNA sequencing, so that we can match each perturbation up with the overall effect on the transcriptional state of that cell. So I'll just go through each of these technologies and show you how they're informing our design of next-generation cell therapies. So a couple of years ago, Eric Schifrit was a postdoc in my lab. Julia Carnevale, who's an oncologist, was a postdoc in Alan Ashworth's lab at the Cancer Center here at UCSF. Actually, I should mention Eric and Julia have now both gone on to their own labs. Eric has a lab in Tel Aviv, and Julia, we recruited to the Genomic Immunology Institute here at UCSF. But they teamed up a couple of years ago to bring genome-wide screens into primary human cells. Genome-wide CRISPR screens had been used for a number of years at that point in cell lines and in animal models. And the trick was that we could make stable Cas9 expressing cells. We didn't have that luxury in T cells. We're having a lot of trouble getting Cas9 stably expressed in T cells for reasons that we didn't fully understand at the time. We were able to use lentivirus to introduce guide RNA libraries. So Eric and Julia hit on a clever hybrid strategy where they actually introduced a library of lentiviral guides targeting every gene in the genome, and then they electroporated the cells with Cas9 protein. And they found that the Cas9 protein could find the guide RNAs that are expressed from these lentiviral integration cassettes, and even this transient pulse of Cas9 was sufficient to leave the cells permanently genetically modified. And because we used lentivirus to introduce the guides, the lentiviral integration cassettes could effectively barcode which gene had been disrupted in each cell in this population of human T cells. So that technology was published a few years ago, showing that this could be used for genome-wide screens. But Eric and Julia have gone on and really pushed where this technology can take us. They've now done a number of different genome-wide screens, not only to find all the genes that affect the extent to which T cells will proliferate when they're re-stimulated. And the way they do that is by using a CFSE dye, which will dilute as cells divide. So you can separate dividing cells from non-dividing cells. And they've done genome-wide screens under these different conditions that are designed in a reductionist way to mimic different aspects of suppressive tumor microenvironments. So they've done it in the presence of an adenosine mimic. They've done it in the presence of immunosuppressive drugs like cyclosporin and tacrolimus. They've done it in the presence of TGF-beta, which is a cytokine, which can suppress T cells. And they've even done it in co-culture systems where they mix the T cells with suppressive regulatory T cells. And what they found was that they could then separate the dividing cells from the non-dividing cells and ask which perturbations allow the cells to continue to divide even under the suppressive conditions. They could actually start to match up different genetic modifications that conferred resistance to these different immunosuppressive conditions. But what we got interested in were a small number of perturbations that really conferred broad resistance and made these T cells broadly resistant to immunosuppression. And one of these modifications actually conferred resistance to all of these immunosuppressive conditions. It was a gene called RASA2. RASA2, to our knowledge, had never been studied in T cells before. And RASA2 in other cells has been proposed to be a negative regulator of RAS signaling. So we wondered whether we'd actually found the RAS gap, the negative regulator of RAS signaling in T cells. And that actually appears to what would be the case. We found multiple guide RNAs targeting RASA2 that were enriched in the dividing population. That was not true for the other RAS gap proteins, but interestingly, the RAS grip for T cells, RAS grip 1, was enriched in the non-dividing cells. So we really had found this was the known positive regulator of RAS signaling. We found the negative regulator of RAS signaling that we could remove to promote T cell division. And consistent with this idea that this was really the T cell RAS gap that we had discovered, when we actually look at the expression of RASA2, it's preferentially expressed in T cells when you compare T cells to all other tissues in these databases. So we think we found a fundamental piece of T cell biology. But if we want to use this to make better T cells, to remove the brake, one thing we wanted to do was make sure that we weren't causing totally dysregulated RAS signaling. And that doesn't appear to be the case. If we don't re-stimulate these cells and we just let them rest, the RASA2 knockout cells remain CFSE high. They're not dividing out of control. But when we re-stimulate these cells, you can see that these cells are more sensitive to stimulation. They divide more. So we think we're just turning up the gain and making these cells more sensitive to stimulation. And consistent with that, we actually found that these cells, if we make RASA2 knockout cells and expose them to different doses of peptide antigen in an antigen presenting cell, and then look at the phospho-ERK signaling, which is downstream of RAS, we found that we make these cells more sensitive to lower doses of antigen density, and we can actually turn up the amplitude. So we think we've really found this dial that we can turn up the gain and make T cells more sensitive. And consistent with that, when we actually look at CAR T cells, and we teamed up with Justin Acom, who had engineered CD19 positive leukemia cells that express different levels of CD19. The wild type NOM6 leukemia cells can be easily killed by controlled CAR T cells or the RASA2. But as you get to these cancer cells that express very low, almost barely detectable levels of CD19, then the RASA2 cells start to really outperform the control CAR T cells. So we think that at least in vitro, we can make these cells more sensitive to low antigen. So that's desirable. But we worried that if we made these cells too sensitive, they might burn out. And that what was potential is that maybe as the cells were chronically stimulated, if they're so sensitive, they could become dysfunctional or exhausted. So Eric and Julia set up a repetitive stimulation assay where they did 6 rounds of stimulation every 48 hours with cancer cells to see how the RASA2 cells would perform over time in these assays. And we were really excited about this. In fact, it was the opposite. It was the RASA2 cells actually had more durable function. The control T cells, in this case, TCR engineered T cells, recognizing an antigen found on the cancer targets, the control cells started to dysfunction over time, and the cancer cells would start to grow out. But the RASA2 knockout cells, they actually had durable function over time, suggesting that not only were the cells more sensitive, they actually had a more durable function. And that was true not only for TCR engineered T cells, but also for CAR T cells, where CD19 was introduced, CD19 CAR was introduced into the TCR locus. Again, more durable function. So that's in vitro. How does this actually perform in vivo? And so we actually took the CD19 CAR, knocked into the TCR locus. So this is one of the best CAR T cells out there. But if we gave them the CAR T cells at a low dose, these control CD19 CAR T cells failed to control leukemia in humanized mouse models. You can see the leukemia grows out, shown here by bioluminescent imaging at this dose. But with the same dose of CAR T cells with RASA2 knockdown, the cancer, the leukemia control was markedly better, and that translated to improved survival in this model. So we think we've really improved in vivo function of CAR T cells. And that's not limited to just a leukemia model. In collaboration with a group at St. Jude's, we also saw improved tumor control using an F8-2 CAR targeting a solid tumor model, a sarcoma model. And we can see an improved tumor control here with these RASA2 knockout CARs. So just to zoom out, I think we're really excited about RASA2 as one way to make CAR T cells even better, but we're really excited about the power of CRISPR to test every gene in the genome, discover new biology, and find new modes of improving specific behaviors to build better cell therapies. So what I told you about was loss of function, but that we've come to realize is really actually a relatively small slice of all the perturbations that are potentially possible. You can get loss of function of the cells of the genes that the T cells naturally depend on. But we'd like to be able to think also not only about what we can get rid of, but what can we add? And that could include genes that T cells may not even use under these natural conditions, but when you put them on in T cells, might add a desirable property. We'd like to be able to test every gene in the genome to discover which genes could be added or subtracted to T cells to make them more effective. And there's again a tool that's been used in cell lines and in animal models that we wanted to bring into human T cells at scale. And this was CRISPR activation and interference. So again, just to briefly say what CRISPR activation is, instead of using CRISPR as a nuclease to disrupt a genetic sequence, this is essentially using CRISPR as an artificial transcriptional activator, where the nuclease function is mutated, its nuclease is dead, but the CRISPR complex is fused to transcriptional activators so that you can turn on a gene of interest by recruiting this to a target site, transcriptional start site in the genome. This can also be adapted into a transcriptional interference tool by fusing it to transcriptional inhibitory domains like CRAB. So this was a huge technical problem to how to introduce these big complexes stably into human T cells. That had really been something that we wanted to do in the lab for a long time, but had not been able to technically achieve. But two postdocs in my lab were undaunted by really a long track record of technical problems in this field, Ralph Schmidt and Zach Steinhardt. And they took this on head on. And what they found was that by systematically optimizing the lentiviral manufacturing steps and the transduction steps, they could actually get this machinery in very efficiently to T cells with some improved protocols. And so with that in hand, they actually went and took CRISPR activation and did genome widescreen. So they used existing libraries that are publicly available to turn on every gene in the genome. They introduced CRISPR-A stably with a viral transduction, and you end up with a population of T cells where each cell has a different gene transcriptionally activated that you can track through barcodes. And they could have done once you have these cells, you can do any phenotype. They got really interested in trying to learn the rules of genes that control cytokine production inside of T cells. So what they did was they took a population of T cells and they looked in the CD4 cells for what regulated IL-2 production, or in CD8 for interferon gamma. These are just different cytokines. They've now adapted this for a number of different cytokines. But the key is that they actually do intracellular staining for the actual cytokine, so not for a reporter. You separate the cells based on high and low production of the cytokine, and ask which guide RNAs are promoting the different cell states, the regulating cytokine production. And the results were really, really clean and beautiful. And so this got me really excited and has now opened the door to lots of CRISPR activation screens in the lab. So what I'm showing you here and what really caught my attention was scatter plots showing the reproducibility between genome-wide screens done in 2 separate human blood donors. You can see that the results really line up on this diagonal, showing that the results are really reproducible. And this was true for the IL-2 screen and for the interferon gamma screen. The hits make sense. So if you directly transduce IL-2, that ends up in the high bin for IL-2 screen. If you directly transduce the interferon gamma gene, that's in the high interferon gamma protein bin in the CRISPR-A screen. And this was not just true at the level of genes. You could actually see multiple guides for targeting IL-2 or interferon gamma. And you could start to pick out other genes that made sense biologically, or if you turn on activating domains, or you could drive higher levels of cytokine production, or things that dampen T cell signaling could dampen cytokine production. Zach and Ralph actually went on, they did the reciprocal screens, not only using CRISPR activation, but actually used CRISPR interference for the same phenotypes. And again, what you can see is that these screens were really reproducible between independent blood donors. Now, if you transcriptionally interfere with IL-2, that ends up in the low IL-2 bin, and it's likewise interfering with interferon gamma ends up with it in the low interferon gamma bin. So why do reciprocal screens, CRISPR activation and CRISPR interference? What we've seen is that we really get different pieces of information from gain of function and loss of function screens. So for example, if you do CRISPR-I, if you look at the expression of all the target genes that come up as hits in the CRISPR-I screen, what makes sense is that these target genes are well expressed in the T cells, right? So you can only get a CRISPR-I phenotype if the T cell is already expressing the target gene and depending on it. In contrast, the CRISPR-A hits have a much broader range of expression patterns, and some of the strong hits actually have very low or even maybe negative expression in T cells, suggesting that we can find a phenotype by turning on artificially genes of interest that might drive interesting T cell phenotypes. And this was really exemplified beautifully when we look at the CRISPR-A and CRISPR-I hits. And we just look for things that come up as CRISPR-I hits for interferon gamma regulation on the x-axis, or CRISPR-A hits for interferon gamma regulation on the y-axis. And if we annotate these hits based on known regulators of the NF-kappa B pathway, what you can see is that there's a couple of hits that come up in both of these screens, but there's actually a large number of regulators that come up selectively either in CRISPR-A or CRISPR-I. And when we zoom in and see what are these genes actually encoding, it starts to make sense. So a lot of this core intracellular machinery comes up preferentially as hits in CRISPR-I. And I think that's because with CRISPR-I, we're able to hit molecules that are constitutively expressed, that are individually required for regulating interferon gamma production. In contrast, a whole number of genes, including especially these cell surface receptors, were missed with CRISPR-I, but come up as hits with CRISPR-A. And we think that these are not individually required, but are sufficient when they're turned up to modulate levels of interferon gamma production. And you'll see that these encode really important regulators of T cell state and a number of things that are being actively developed as immunotherapy targets, either for drugs or small molecules or for manipulation in CAR T cells. So OX40, CD27, CD40, 41BB all come up as screens. So we think we've really found an interesting set of genes that are worthy of further exploration as immunotherapy targets. Just give you a flavor that these technologies, that we're able to actually not only pick out hits, but actually start to characterize them mechanistically by coupling CRISPR-A technology with single-cell RNA sequencing. So this is broadly known as Perturb-seq from a number of papers that were done in cell lines and in other models. And we wanted to do this with CRISPR-A in primary human T cells. So the idea was we made a targeted library of CRISPR-A perturbed cells, and then we used single-cell RNA sequencing to try to capture the transcriptome of each one of these states, as well as capture the guide RNA so we could know which perturbation was actually driving the transcriptome changes in each of these cells. Zach and Ralph picked out about 70 different targets from their screens, including some controls, things that were both generally positive regulators of cytokine production and negative regulators of cytokine production. And they made a library for Perturb-seq. And I'll just show you that we're able to actually do this in actually human primary cells from different human blood donors, you could see that we're able to map a number of different cell states in UMAP space. And then we can annotate which are these cell states. These are T cell states that were found in this general population of perturbed cells. And you can actually start to map these cells for interferon gamma, but for other states that are recognizable, and then ask which guide RNAs are overrepresented in each of these clusters that we discovered in single cell space. And this allows us to really start to make a functional map of genes that when we turn them on, move the cells to a certain state. And so looking forward, I think that as we get more and more information about the single cell state of what an ideal T cell will look like for a cell therapy for cancer or for other states, this will verify to us that we're actually finding genetic perturbations that are moving us towards that desired state, and will accelerate the timeline to confirm that we've actually come up with desirable perturbations to manufacture better cell therapies. I'm going to take one slight tangent here into something that I think is opening up some really fundamental tools for probing the genome in human T cells. So one opportunity that comes up by now using, and this is unpublished work, by using CRISPR activation and CRISPR interference, these turn out to be really powerful tools for exploring not only coding, but also non-coding parts of the genome. And so we got really interested in, could we discover non-coding elements systematically using, in this case, CRISPR interference? This is work that was led by Cody, an MD PhD student in the lab. And he got really interested in this locus, which encodes three really important co-stimulatory receptors: CD28, which is a co-stimulatory domain; CTLA4, which is actually a checkpoint, a co-inhibitory domain; and ICOS, which is another co-stimulatory receptor. These three genes arose from gene duplication, and the relative level of all of these genes needs to be tightly regulated to shape the state of T cells. And we know that because there's genetic variants in this locus that contribute to a range of different autoimmune diseases, and because drugs have now been developed against CTLA4, where we can actually mimic CTLA4 to treat autoimmunity or block CTLA4 to treat certain types of cancer and release checkpoints. So Cody wanted to see, how is this locus actually regulated in different primary human T cells to get the appropriate balance of these different receptors? And this was something that we didn't really have the tools to do until just a couple of I guess, about a year ago. And what Cody did was he took a library of guide RNAs that densely tiles across this whole genomic region, and he introduced them into T cells, either conventional CD4 cells or regulatory T cells that have different expression patterns. And he sorted the cells for high or low levels of each of these receptors using FACS. That allowed us to find guide RNAs that actually target elements that tune the levels of these different gene products. So we did this for CD28, CTLA4, and ICOS. And I'll just zoom in on the CTLA4 to give you a little vignette of how this is starting to teach us about how non-coding elements are actually used. If you look at the CTLA4 region, we actually are able to find a non-coding element in stimulated conventional cells, where if you bring CRISPR-I, you actually block the ability of CTLA4 to be turned on properly. That lines up with something that has chromatin features suggesting that it can act as an enhancer. We can actually now link this enhancer selectively to CTLA4. And we can actually, by doing this experiment not only in the conventional cells, but also in the regulatory T cells, which have a specialized role in preventing autoimmune disease, we actually found that the regulatory T cells use a different element to constitutively keep CTLA4 levels high. And so regulatory T cells depend on this element for their levels of CTLA4. So we start to actually work out a code of different cell types and conditions that depend on different non-coding elements. And this becomes relevant for interpreting disease genetics because there's actually a lot of linked genetic variants in this locus. This is the index SNP over here, but many of the variants in this region have high degrees of linkage disequilibrium. It's been hard to sort out which ones are actually affecting cell state, but this allows us to start prioritizing some candidate variants that might be affecting these cell type specific non-coding elements and set the stage for future studies. So I think I'll pause here for 1 second and just reflect on what I talked about before I show you one last story of largely unpublished data. What I've tried to talk about is really a set of tools and approaches that allows us to decode these cell circuits that control T cell behaviors. We think a lot about these models where there's both coding genes and enhancers that work coordinately to shape how T cells respond to stimulation, how they move into different states to be compatible with immune responses, including clearing tumors. And now we can systematically decode these elements and find which ones are important for controlling cell behavior. And this has huge potential to teach us how these cells are wired, to find new targets for drugs, but also to inform design of future cell therapy. But where I see this going is, I think that these screens, which are now able to turn on and turn off every coding and non-coding element in the genome, they're teaching us the basic vocabulary of genetic elements that are important for controlling T cell function. The next step is to think about how do we actually take that vocabulary, take that lexicon, and actually start writing synthetic programs with it to have a new level of control of shaping how T cells will behave. And so that brings us really into an era of synthetic biology. Can we write sequences that don't exist in nature, that have complex programs that make cells behave how we want? And I'm going to talk about work that's been started by Theo Roth, a former MD PhD student in the lab who's now at Stanford, and Franzi Bleschka, who's training as a pediatric oncologist and as a postdoc in the lab. And what a couple of years ago Theo showed was a new way of being able to take a whole pool of different DNA sequences, knock them all into the same targeted site in the genome, and then race these cells against each other and see which of these knock-in sequences has driven a behavior of interest. And what this means is that these sequences, which now can, with our latest technologies, can be many KB, we can actually start thinking about writing a string of sequences, for example, polycistronic genes or even other kinds of codes that don't exist in nature that we put into a defined site in the genome to see if we can drive T cells into desirable states. And we're still exploring what this is capable of, but I'll just show you some of our unpublished work. What Franzi and Theo have now done is develop a modular system. So they take a homology-directed repair template, and they have different modules to allow very rapid cloning of different libraries. So they have one site where there's a functional module site where you can rapidly clone in libraries, for example, of 100 different transcription factors into this site, or over 100 different cell surface receptors, including some that don't exist in nature. So this includes synthetic chimeric receptors, where you fuse activating and fuse extracellular and intracellular receptors in novel configurations. So you could take different functional modules and combine them with different TCRs or CARs that have different abilities to direct T cells against different antigens. And this is just the tip of the iceberg of the different type of combinatorial design that we could do with this type of template. But the key is that you end up with a large library of different templates that get knocked into a defined site in the genome. You end up with a pool of different cells, and then we put these cells through many different challenges to pick out which are the actual sequences that allow the cells to have fitness advantages in different contexts. And so one of the contexts that we've cared a lot about is how cells will respond as they're chronically stimulated. So this is very similar to what I showed you with Eric and Julia. So now we're able to actually pick out cell surface molecules or transcription factors that increase the fitness over multiple rounds of stimulation. You can see, for example, knocking in the IL-2 receptor, IL-2RA, tends to increase the fitness of cells, and that fits with some known biology. But we're actually also able to pick out transcription factors. One that's been in the literature from Patrick Hogan and Anjana Rao is that BAD-F can increase T cell fitness. We found TFAP4, which there's some literature, but less. It wasn't one of the obvious ones. But TFAP4 had a strong effect on increasing cell fitness, not initially, but through chronic stimulation. And we're actually able to start to piece together transcription factors or synthetic receptors that give different either CARs or TCR engineered cells benefits under different specific challenges. And one of the things that we cared about a lot was that we looked at a specific CAR that Crystal Mackel a few years ago showed can fail because this has a tonic signaling. This is a high affinity CAR targeting a GD2, and it chronically signals leading to T cell dysfunction with certain features of exhaustion. We found that TFAP4 has a big effect on driving increased fitness in these tonically signaling GD2 CARs. And that translates when you put TFAP4 into these GD2 CARs to improved cancer control. And in vitro, that was also true for CD19 CARs. That also translated to improved control of GD2 positive cancer cells in vivo. So you can see improved tumor control with these TFAP4 knock-ins and improved survival. Now, I'll just show the last thing I'm going to show, the last piece of data, is that this modular cloning strategy has one other feature. It can actually allow for combinatorial cloning. So we could actually take our 100-member transcription factor library and put it in tandem side by side to end up with what ideally 10,000 different combinations of different transcription factors that you could form 100 by 100. And we put this into a template that encoded the GD2 targeting CAR, these tonically signaling CARs that can fail from dysfunction or exhaustion. And we just saw which combinations of transcription factors now increase fitness in this particular GD2 CAR. And this was powerful because we know that many times transcription factors don't work in isolation, they work coordinately. So this showed us a couple of things. First of all, if you just look at the representation in the plasmid pool, you can see that we're hoping to actually knock in things that go up to almost 8 kb in size. And in fact, we were able to see reasonable representation of these knock-ins that go up to almost 8 kb, suggesting that we're now able to do pooled screens with really big templates. And when you look across all of this, we actually start to see some clear winners. And this combination of BADF and TFAP4 really stood out as a winner. So we wanted to see, is that combination actually better than the singles? So Franzi actually went back and tested this head-to-head, knocking in a construct with the GD2 CAR, along with BADF and TFAP4, or just knocking in control with a single transcription factor, either TFAP4 or BADF. And in these mixed assays for competitive fitness, you can actually see that the doubles actually outperform and have a competitive fitness advantage. And that translated to better cancer control in vitro and in vivo. So I think I want to just pause there and say, we're really excited about some of these constructs that are coming out, but even more broadly, we're able to now think about designing synthetic biology sequences at scale. And we've gone from dozens to hundreds, now up to thousands of different synthetic sequences that we can introduce into cells to start to learn rules. And I think that every aspect of that's important for designing cell therapy can now be tested at scale. You can start thinking about what are the actual receptors that you want. How do you actually design the linker lengths, non-coding sequences that turn on these features? So we can really start playing with every aspect of cell therapy design at scale. And the last thing I'll just show is not data, is thinking about, well, I've sort of put out a big goal of thinking about how do we program cells to understand them and think about driving a whole new generation of cell therapies. Obviously, that's bigger than any one lab. And we've put a lot of effort into bringing together labs at the Gladstone Institute, which is a nonprofit research institute in Mission Bay, and at UCSF, trying to bring together a community here in the Bay Area to systematically bring together different disciplines that are required to tackle these big goals. So we're thinking broadly about many different, both healthy states and disease states. How do we understand how cells are failing? And how do we use high throughput screens, which I laid out today, to really understand the rules of different behaviors that we care about in immune cells? Where do cells traffic in the body? How strong will the responses be? What do these cells recognize? How long will they endure? And how do they interact with the tumor microenvironment? And once we use these high throughput screens, how do we take that lexicon and translate it into improved synthetic biology design? And then go all the way into clinical process development to make synthetically programmed immune cells, and then go into clinical trials to learn the lessons about what works and what doesn't work and continue to iterate on this. So this is a slide that has a lot of information, but it's a group of us that have been working very closely, and we're nucleating a bigger community around genomic immunology. And we're thinking about, how do we accelerate the discovery engine to think about how are we going to make cells that are programmable in what they recognize, that can be tuned based on the receptors, how strongly and in what way the cells will stimulate. How do we make cells that not only go to a desired site, but actually productively interact with the endogenous immune system to get coordinated responses? And I think one of the big goals that we have is, and this is still a bit of science fiction, but not that far, is can we use immune cells as a drug delivery device? Can we make immune cells that are programmed to go to a particular site of pathology, sense that pathology, and in a local and tunable way, deliver some therapeutic molecule to that site? And so I think that puts us in a range where we can start thinking about treating not only cancer, but also about autoimmunity, infectious disease, and maybe making synthetic programs that'll make immune cells that treat cardiovascular disease and neurodegenerative disease. So I think we're in the early days of imagining what's possible. I'm going to stop there. I'm going to thank the people in my lab who have done this work, and also highlight that this was done. Everything is done in a broad collaborative network, and we really benefit from a broad network of funding sources. And thank you again for having me today.